This is a special episode of the Immunology Podcast, IUIS 2023, Day 5, the last day. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. Today we're back with our final episode from IUIS 2023 in Cape Town, South Africa. We'll be discussing highlights and sharing our thoughts on how it all went. If you're tuning in for the first time, be sure to visit www.immunologypodcast.com or your favorite podcast app to check out our episodes covering the first 4 days of the meeting. We're going to kick things off in just a minute, but before we get to that,
join us for a live webinar on January 23, 2024 to learn more about the most recent advances in CRISPR genome editing of primary cells. Dr. Zhen Zhang from the Perelman School of Medicine at the University of Pennsylvania will present his findings on the development and application of an engineered peptide-assisted genome editing CRISPR-Cas system for efficient editing of primary cells. To register for the webinar, please visit www.stemcell.com/Dr. Zhang webinar.
pen, add it again.
Wow, I'm signing up for this. For sure. Well, you know what I'm signing up for too? Last day of IUIS 2023. Is it?
Yes.
We've reached the end.
We're done. Stick a fork in us.
Yeah.
Is that an expression in any of your other languages? In
No.
English, it's like stick a fork in me, I'm done.
No.
No,
That
you don't have
sounds
that.
disturbing.
So
Yeah. Well, we are both here already. Jason is rocking an Immunology Podcast t-shirt.
yeah, we're gonna have to do a twinsy picture after this and put it on the socials.
Definitely.
All right. How was the morning keynote?
Oh, it's really good. We had Timothy Springer from Harvard Medical School. He is, of course, institution when it comes to understanding integrins. And he gave, I think it's been a kind of a theme for the keynote speakers, kind of a lecture on the discovery of integrins and the role he and his lab played in it. He showed one of his own pioneering work identifying LFA1 as important for the immune synapse, and many other contributions of his lab and other people kind of build up on our understanding of integrins. And he finished up with a really cool data, single molecule analysis that really shows the mechanism of activation of integrins at a molecular level. And he came about this under kind of saying it's important for integrins that they increase the intensity of the immune synapses that contributes to recognition, but they mustn't sacrifice specificity. And that's kind of an important concept to keep in mind when talking about these molecules. And this is in a way solved, the way that nature solved this is by having these different conformations of integrins that get activated only when they're accompanied, for example, by TCR stimulation. So therefore, there's these different conformations, and he shows a lot of kind of single cell data, a lot of really cool analyses of how do we understand this dynamic to work. And he finished the talk with a video of some kind of contemporary dancers interpreting how integrins work, and it was really cool to see. I wish I had filmed the whole thing, but yeah. So really nice talk. I have to say I don't think about integrins often enough, so I really appreciated the overview.
But I think about integrins a lot because I have anti-integrins coursing through my blood on a daily basis. Yeah, so but they're near and dear to my heart because I don't have a good relationship with my integrins.
Well, he did mention, he did show how much of a therapeutic targets they can be.
Oh, and Entyvio is a fabulous drug.
Okay.
All right. Well, I went to a talk on immune regulation, which is really a talk about multiple talks about STAT3.
Oh, yeah.
So sometimes there's a theme that subdevelops that you don't realize on the title of the session. So I had first up was Mohammed Rida Barbouche from Bahrain. He really demonstrated that. So what was interesting about this, they were using a lot of human clinical data. Mm-hmm. And there's a lot of STAT3 related inborn errors of immunity that are known in the West, but he found one that was actually more common, that's PGM3 deficiency, and more common than STAT3 deficiencies, but has similar phenotypes. Mm-hmm. And he was able to establish that has impaired signaling through GP130 glycosylation deficits, and that this then signaling deficit has this IL-6 signaling deficit, which leads to an IL-21 deficit production, this weird IL-21 production, upregulation of IgE class switching. And so it was just interesting that in this other cohort of people, you had a different gene signal than you would usually anticipate. That provided a lot of learning. And then Cindy Ma came on and talked about the role of STAT3 in human immunity, and talking about, there's lots of these inborn errors, and there's this hyper IgE syndrome, which can be autosomal dominant or receptive. Autosomal dominant is known by caused by STAT3 hyperactivation. And so what happens is there's this B cell extrinsic deficit due to T follicular helper formation, but a memory B cell intrinsic in STAT3, but notably not STAT1's required for the memory B cell function for IgA and IgG secretion. And again, this is an IL-21 mediated process, like the story from Mohammed's lab. And so there was a little bit of a review of some kind of current level work on STAT3, hyperimmunity, dysregulations in immune system, not so much, chronic immune deficiency, but hyperactive systems that are based on, at least a lot, by STAT3 being screwed up. So that was my first talk.
Okay. I went for the morning session. I went to this really nice reproductive immunity talk. So it started with Dr. Rana Chakraborty from Mayo Clinic. And she spoke about the role of Hofbauer cells, which are kind of a placental, I think, macrophages, and how talking about how sometimes they get infected with CMV, and how CMV can potentially increase susceptibility of these cells also to HIV infection, and this can be a way of vertical transmission to fetus. And then after her, we had Melanie Conrad from Charité. She was talking about So I think the bulk of her work was about antibiotic use in pregnancy. She also mentioned a little bit about, she had some analyses about depression in pregnancy and the COVID pandemic. I think I missed a little bit. I think that the theme was stress during pregnancy has negative effects, and her kind of disease of model disease is asthma, and I think she wanted to kind of convey that stress with pregnancy can also increase things like asthma in the progeny. But I kind of missed a little bit that part. But what I was very clear, was really nice, was her work on the effect of antibiotic use in pregnancy. She had the systems in which she had data on pregnant mice treating vancomycin. She also made the point that they were treating these mice daily, and they had figured out that by giving the vancomycin within a very kind of sugary syrup, the mice actually took it very willingly. So I think, I don't know if other people are struggling with getting their mice to get their antibiotics. She was very pleased with herself and their system. So she had data showing how dysbiosis caused by vancomycin shifted ILC populations in the gut and activated, and this resulted in LPS translocation activation of Th17, and how this resulted in asthma and sensitivity in the lung of the pups. But I think what was also really cool is she talked about the idea of like how can you prevent asthma? And she talked about, this idea that farm antigen exposure can help, like, it's kind of a good thing. And so they kind of pinned down on one particular strain of bacteria that they show that it would generate, not infection necessarily, had a model in which they were just exposing the mothers with dried bacteria products. And that increased a production of IgG1, which protected the pups. So also really nice, kind of some more direct evidence of the protective capacity of certain bacterial strains. And the session was finished by Tamara Tilburgs from the University of Cincinnati, who did really nice session about mechanisms of tolerance of fetus. She particularly honed in the expression of HLA-C and HLA-G on trophoblasts, which are not found in mice. So it's really hard to model this in mouse. but how these cells can induce Tregs that protect the fetus. She did some, she showed some more mechanistic data, and she also talked about specific subsets of CD8 T cells in the decidua. So overall tolerance, and while how you get tolerance while still protecting the fetus from infection. Really, I think it's such an important type of work.
I kind of tie in a little with my next one, which was on mucosal immunology, but had a lot of fetal work in it. So there was a really cool talk by Tim Hand. And he starts by recapitulating some things that they're well known if you're a microbiome nerd, which is early microbiome disruptions in infants and children can lead to disease, antibiotics in the mother or the baby at birth or young age. One of these is necrotizing enterocolitis, which is super terribly bad, can lead happens in premature infants, can lead to intestinal resection and death. It's super bad. The intestinal resection can have problems later in life. What's known is that Bifidobacter and Clostridia take longer to engraft preterm in people that are premature, and also the body can't but the body also, because the gut isn't mature enough yet, it doesn't respond properly to those when they start coming in, and so that's a dual-edged issue. So just giving babies Bifido and Clostridia may not be okay, because what they start out with is Enterobacter, right? And so the maternal IgA is important for preventing NEC, and that's also known. And so they were able to start doing some maternal IgA studies through donor milk and other things, and they found that milk that could prevent NEC was better at binding IgA. And they also found that mothers' properties of their milk doesn't really change over time, at least during the time course. So if you have whatever's in your milk is going to be in your milk, and so you're either a good responder or not. It doesn't matter if your kid has something going on or next pregnancy, it just persists. So kind of your phenotype for producing what your IgA you produce seems pretty locked, which was interesting. They also demonstrated that then they did a really cool study where they crossed IgA knockout mice to wild type mice. And so the pups always were IgA competent, but then you would either have a mother or father who couldn't produce IgA. So if the mom couldn't produce IgA, right, during weaning, you have no IgA in your milk. So it's a really good controlled experiment. And they demonstrated that IgA in the milk regulates intestinal epithelium prior to weaning and leads to early maturation in secretory lineages. And if you could transfer the microbiota in the knockouts, the knockout transfer, so knockouts led to earlier secretory lineage maturation, right? Because they're like, the intestine has to deal with this bacteria not coated in IgA anymore. And then that has this Th17 response phenotype to it with these ROR gamma T cells, and transfer the microbiota from one side to the other will induce this phenotype. And then furthermore, these ROR gamma T cells persist into adulthood, and so they can find it way later. And they have very modest effects, but they could take these pups that were from IgA negative moms. They have worse indomethacin colitis as a result. Not a ton worse, but a little worse. And more IL-17, but P was only 0.06. But the point is, this is an effect in an adult that was based on what they had as breast milk as a baby.
Wow.
Nothing's changed since then, right? They're IgA competent. They have no genetic issues because they're haplotype, they're plus minus, they can make IgA. They're seeing this effect. So this is really cool. They're trying to figure out how to start making this into some type of therapeutic, but there's a lot of work to do because making monoclonals to IgA is hard. And then another person who says early investigator award was Roland Ruscher, who was looking at mechanisms to deal with pediatric and adult onset colitis, and they couldn't talk a lot about it because they're working with a company called Macrobiome Therapeutics, but essentially that company synthesized a hookworm-derived recombinant protein, and hookworms generate tolerance, and that's thought to be, and we know that people with hookworms have less inflammatory bowel disease, and so they're applying that at least in mouse models, and it makes the IBD go away.
All right.
In the mouse models, so that they're working on that, and that's what I got.
Okay. Well, for the last keynote of the conference, we had Sarah Gilbert from University of Oxford, who is she is very well known for spearheading the effort of the vaccine, the COVID vaccine that University of Oxford made, this adenoviral vector vaccine, and so she talked about. She clearly has been involved in this for many, many years. So it was such an inspiring talk about, what it takes to make, to test, and to distribute these vaccines against, pandemic. She talks more about epidemic diseases or emerging diseases. She started her keynote talking about her experience trying to test a vaccine against Ebola during the 2014 outbreak. And I thought what she was very keen on showing is that although they had the vaccine, they took so long to come up with the right ethical way of testing it in the field that it took 4 months for the clinical trial to be approved and the design to be approved. And by that time, the peak of the epidemic was over. So she really kind of wants to bring home the message that we need to figure these things out before we need them. And I think that was an overarching topic, a theme in her presentation.
So that's really interesting because at least in cancer trials now, right? If you're going to die from the cancer, the threshold for the trial has to be, is it less likely to kill you than the cancer?
Well, but for the Ebola, because you could consider that it was unethical not to vaccinate everyone just in case. So that's basically they had to, but if you vaccinate everyone, there's no comparison that you can do. So what they did is that they compared two groups. They were in a way divided in a group that got vaccinated now and a group that got vaccinated in 3 months. I don't know exactly how that division was made, but there were basically two comparisons. And you could see where there was, if the vaccine was good enough, you would see that in the.
That's interesting. I mean, I could also argue just vaccinate everyone because Ebola is almost universally lethal, and so.
Well, I don't know. Well,
yeah. Anyway. It's interesting.
So, and then she continued. She talked about, mentioned other kind of epidemic viruses, Nipah, Hendra virus, Lassa fever virus, how different approaches to different viruses. And then she also talked extensively about her experience bringing over the SARS-CoV-2 vaccine, how important it was to design, all the details in the vaccine. She talked about starting from the sequence design, going over to how they decided upon the amount of doses and how they managed the time between doses, how they initially wanted 4 weeks, but then because of the availability of the vaccine and for a public health reason, they extended to about 12 weeks, and that actually ended up being better at protection. After the two, this is of course the AstraZeneca, so they teamed up with AstraZeneca for making this vaccine. So this is the Oxford AstraZeneca chimpanzee adenoviral vector vaccine. Yeah. And so she talked about that and how important it is to really have these things planned out. And of course her experience, speaks volumes about this.
Yeah, I never understood why it was 4 weeks for COVID to begin with, because you usually almost never do vaccines 4 weeks apart, even for respiratory. And respiratory stuff usually have a month or two of protection.
I think it was I think it was just to give the shortest time possible for people to have the two doses that were considered protective. And that was the shortest thing that made sense.
Yeah, okay. So if you're going for as close together as possible, 4 weeks is the answer.
Get results as quickly as possible. Yeah. But then a lot of people got vaccinated longer than that.
Yeah.
So I thought that was really cool. And I just want to make a quick, before we leave for today, yesterday was Diane Mathis' presentation. She won the Menarini Prize for outstanding woman immunologist, and she's really well known in the field of immune regulation. Her team discovered the role of air in the antigen presentation in the thymus. So big, very important discovery. And also a lot of other things. Visceral adipose tissue Tregs were also discovered in her lab, and that opened floodgates to a lot of things. And she has a lot of research, so I just want to say it was really nice. Her presentation was mostly about her most important contribution, or her more surprising contributions, I guess. But yeah, congratulations to Diane Mathis on that.
That was cool to see.
And you know what? Today I signed my first autograph. Can you believe that? What? I signed an autograph on the back of a 50 Rand banknote.
Is that a crime in the?
I don't know. About $2.50. So if you're listening, Casper from Berlin, your colleague will bring you that banknote hopefully when he comes back.
There you go. Well, dang. All right. Well, that brings us to the end of our IUIS 2023 episode series. We've met a lot of people and had a lot of fun. Don't forget to subscribe to our newsletter at www.immunologypodcast.com. You can also reach out to us on Twitter or X or whatever it's called at Immunopodcast or via email at info@immunologypodcast.com. See you next time.